Didn't lose anyone. Okay. Hello. As chair of the Plant Conservation Federal Committee, I'm pleased to introduce our two speakers today. Dr. Brian Irish, as a geneticist, curator, and lead scientist with the USDA Agricultural Research Service Plant Germplasm Introduction and Testing Research Unit in Prosser, Washington, is our first speaker. He obtained his Bachelor's of Science in Horticulture, an MS in Plant Pathology, and a PhD in Plant Science from the University of Arkansas, and spent the first 11 years of his ARS career at the USDA ARS Tropical Agriculture Research Station in Puerto Rico, where he managed clonally propagated, agriculturally important tropical genetic material. Missing a word there, sorry. Now the project he leads is responsible for the acquisition, maintenance, disease-free propagation, and regeneration, characterization, evaluation, and distribution of nearly 13,000 accessions within the temperate adapted forage legume germplasm collection. For the past 2.5 years, he has also led an interagency BLM ARS project on the long-term conservation of U.S. native plant species used for restoration and agricultural purposes, which will be the topic he discusses with us today. Our second speaker today, or our joint speaker, is Dr. Stephanie Greene. She's a supervisory plant physiologist with the USDA Agricultural Research Service at the National Laboratory for Genetic Resource Preservation at Fort Collins, Colorado. Dr. Greene was educated as a plant breeder, receiving her BS and MS from the University of Idaho and her PhD from Kansas State University. She has worked in the National Plant Germplasm System since 1992 and curated the temperate forage legume collection until 2014 when she transferred to Fort Collins. The National Laboratory for Genetic Resource Preservation provides secure backup storage of the USDA National Plant Germplasm System collection and other seed collections from around the world, making it one of the largest seed repositories in the world. As the seed curator, Dr. Greene is responsible for receiving, processing, packaging, storage, and distribution of the seed, including managing the long-term preservation of seeds of success germplasm. Without further ado, I believe we'll be turning it over to Brian first. Brian, are you able to grab the screen?
So I've unmuted myself and I turned my video on, and I'm going to try to start sharing my screen. Can you hear me, Patricia?
Yes,
and
I can.
You see me? Okay, give me one second. Let's see if this works. And please tell me when you start seeing the title slide of my presentation, of our presentation.
It's there. Yep, looks great. Thank you. Take it away.
All right. Well, as Patricia just mentioned, today my colleague and I, Dr. Stephanie Greene, will be giving a joint presentation on ARS's National Plant Germplasm System's role in native plant conservation and use. So I meant to say good afternoon to most, and good morning if anybody else is joining from the Pacific where I am, Pacific Coast where I am. Thank you for this opportunity to Patricia and the organizers and the PCA, the Plant Conservation Alliance Committee. We were asked to give this presentation by Dr. Pete Vadas, our national program leader. In a few of the announcements early on, we noticed that the title mentioned that we were going to give the presentation on behalf of our agency, and that's not really true. What we're doing is a little bit more focused and, again, giving the presentation on behalf of the National Plant Germplasm System. So our presentation will follow this general outline, with me giving a brief background on the agency, Agricultural Research Service. Then we'll provide a few definitions and justifications. Then we'll focus a little bit more on NPGS's activities related to native plant genetic resources conservation and utilization, followed by a shift in gears and a little bit more focused discussion on Seeds of Success, the Seeds of Success collaboration, the Bureau of Land Management long-term collaboration that is implemented predominantly in two different sites, the one that I'm affiliated with in Pullman, Washington, and the one that Dr. Greene is affiliated with in Fort Collins. So that's about 2/3 of the way through the presentation. Dr. Greene will take over and we'll discuss Fort Collins activities as well as a summary of the presentation. We thought that before we got too far, we would say that the National Plant Germplasm System is invested in native plant conservation, and consistent with its mission and role, the Npgs has, can, and more than likely will continue to contribute to conserving U.S. native plant genetic resources or plant germplasm. And so also many of the activities that you'll hear from today in our presentations align, and many of our activities along with native plants align well to the National Seed Strategy, which ARS is a member to. So information from this slide was taken from our ARS official About Us website. And so the Agricultural Research Service is one of 17 or 16 different agencies of the Department of Agriculture. It's the principal in-house agency that conducts research in order to deliver cutting-edge scientific tools and innovative solutions for American farmers, producers, industry, and communities in order to support the nourishment and well-being of all people, to sustain our nation's agroecosystems and natural resources, and to ensure the economic competitiveness and excellence of our agriculture. If the audience would notice on that second bullet, I've underlined two terms, and that's because this is an area that we potentially can continue to collaborate with members of the PCA, and this is where the National Plant Germplasm System fits in within the ARS. Stephanie and I both thought that it would be appropriate to define a few terms before we got too far into the presentation. I know I've already used that first term, and that's germplasm, or more specifically plant germplasm, or plant genetic resources, abbreviated PGR. These are the seeds, fruits, cutting, pollen, and more, and is the raw material that underpins food, fiber, forage, fuel, flowers, and I wish the word restoration started with an F, but restoration activities as well. So the other term that will appear throughout is plant accession, and this is the plant material from a single species or taxon collected at one time from a specific location, while capturing the diversity present in that plant population. So in gene banking, the term that we use is accession, and I understand that plant conservationists, especially those that are affiliated with the Seeds of Success program, use the word collection. We would use the word collection in the field, but once the germplasm is incorporated into a gene bank or into the NPGS, we use the term accession. So I don't think I have to convince many in this audience that plant germplasm or plant genetic resources are key. And they are key in meeting threats to global agricultural productivity through continued progress in agriculture and in plant sciences. It's also the plant breeders and other scientists that need to be able to access continually this genetic diversity, and that this genetic diversity is predominantly sourced from plant gene banks, like the National Plant Germplasm System. The USDA's National Plant Germplasm System is an organization of 20 different sites that are located across the nation in both continental and insular regions. The two sites that the site that I'm affiliated with in Pullman, Washington, and the site in Fort Collins, Colorado are integral, and you'll hear quite a bit more about these two sites during our presentation. So many of these sites have long-standing partnerships, and in several cases are co-located with land-grant universities, as is the case in Pullman and in Fort Collins. Also might have other affiliations with state agricultural experiment stations, or even partnerships with other commodity groups and the agricultural or horticultural industries in those regions. If you can think of an important agricultural crop, the National Plant Germplasm System more than likely conserves plant genetic resources for it. This slide summarizes some of the holdings for the National Plant Germplasm System, and it is one of the largest, if not the largest, gene bank system in the world. It currently conserves about 600,000 accessions, or just over 600,000 accessions, of 16,000 plant species. And the bar graph to the right, it shows the gain in number of accessions over a 10-year period starting in 2010 through 2019. These are large collections of major staple crops important to the US and to the world, but also specialty crops or even collections of important plant species that are not held elsewhere in the world. All of the information that is associated with these accessions is managed with a specialized software that is dedicated for and it was developed specifically for gene banking in an international collaboration between USDA, ARS, Biodiversity International, with support from the Crop Trust. And this software is called the Germplasm Resources Information Network, or Greene Global. And as I said, it's an international standard that is being used by the Npgs as well as many other gene banks around the world. I know this is a screen capture of a very small font, and I don't think the point is to pay attention to the details here. It's just, again, a screenshot of the Greene Global software that we as gene bank managers use to enter information and to manage information associated to the accessions that we conserve. It is called the Curator tool. And this is where it's a relational database with many different tables that, again, we used for managing information associated to the different accessions. There's information on taxonomy. There's information on passport, the history of where the germplasm originated. There's information associated to evaluations or distributions in this relational database that we use. And so there's these two interfaces. This is the interface that we as gene bank managers use for inputting information and managing it. The other interface of Greene Global software is a browser-based interface where stakeholders or the public can access information about the same genetic resources that we manage online. And so again, with a browser, one would navigate to this Greene Global webpage and search for information about specific germplasm. I meant to say in that previous slide there was a highlighted Latin binomial. It's also highlighted on this one, and that's Achnathorum hymenoides. And that's an important grass species that's used as a forage, but also important in restoration activities that is mostly found in the Western US. And so I've highlighted it here and in the previous slide and in a few more slides, just to make a point about native plant genetic resources and using that species. So again, a stakeholder would navigate to this page and search using the different features there. There's an advanced features that will help you filter on the locality or origin of germplasm or different aspects to be able to home in on specific germplasm that then they could potentially request if they become a registered user and provide a justification for that need of that germplasm. This is a color-coded slide on purpose that I've adapted from National Program Leader Dr. Peter Brading, and it outlines these major phases of plant genetic resources management. And as I said, it is color-coded on purpose because those activities on the left-hand side of the screen are of higher precedence and therefore using those red or hot colors compared to those activities on the right-hand side. And if you notice, the word maintenance is underlined, and that's because it's very important that we do everything we can to conserve the close to 600,000 accessions that are currently held by the Npgs. In order to do that, one of the important activities is regenerations. Regenerations for seed propagated plants and means just that, to increase seeds. For clonal propagated crops, it might be that the material needs to be regenerated because of diseases. And so as seed stocks run low, for example, or because we're distributing them, or as the viability declines below a certain threshold, these accessions need to be regenerated. More on that in a moment. So I just mentioned about Greene Global and how important it is to manage all the associated information with the accessions, it's important to understand what it is that we are holding. The word acquisition is two-tone, and that's on purpose. That's because we've gone through a major acquisition phase over the years, and we've sort of getting to the point where we have to be very, very strategic in the NPGS as we add more germplasm. And so if there's gaps to be filled or germplasm that is threatened, this is a reason for adding them to the collections or acquiring that germplasm. We also are a service-oriented organization where we would like to get germplasm to the stakeholders that need it, and so distribution is also very important. The priorities on the right-hand side are where a lot of extramural agreements might come into play, or collaborations, where we can spend some additional resources in characterizing or evaluating plant genetic resources, or even in some aspects of plant breeding or enhancement, and in any other activities that support these preceding priorities. So most of the genetic resources in the NPGS are held ex situ in the gene banks on the map that we saw. And so much of the germplasm is also introduced, but there is a significant proportion of native plant genetic resource conserved, to the tune of about 43,486 accessions in 6,000 taxa and 174 families. And even though that's only about 7% of the total proportion of accessions in the NPGS, this is still significant. And of that 43,000, about 44% has come through the Seeds of Success program that we will discuss a little bit more. So the emphasis for the NPGS has been to acquire native plant genetic resources that are crop wild relatives or wild utilized species that have potential ornamental, medicinal, forage, or restoration values. And to give you an idea of the activities of the number of distributions, in the past 5 years, the NPGS has distributed just under 6,000 orders or requests of 50,000 seed packets of close to 14,000 accessions. And this is true for the native plant germplasm, but also for all germplasm in NPGS. We distribute small quantities in support of breeding, research, and education after a justification is provided for that need. I just mentioned that most of the germplasm is conserved ex situ on site in those gene banks, but also there's efforts underway to complementary conserve through partnerships in situ plant genetic resources. This is a slide that summarizes some of those efforts led by the Plant Exchange Office of the NPGS. And it discusses in situ conservation of cranberry crop wild relatives in partnerships with the U.S. Forest Service, ARS obviously, and the University of Wisconsin. That map to the top right shows some of the 15 national forest sites that are being considered for in situ reserves of these different vaccinium or cranberry crop wild relatives, the populations of these species. It is that same Plant Exchange Office that also supports plant germplasm explorations and collecting trips. These plant explorations and collecting trips happen internationally, but nationally as well, and focuses again on crop wild relatives. The list in the middle of the slide gives examples of some of the collecting trips that were held mostly in 2019, although in spite of our global pandemic, there was some collecting efforts that happened in 2020. And specifically in 2020, those were for Helianthus species or crop wild relatives of sunflower that appears to be collected in Louisiana as well as in California, as well as potato wild relatives in the southwestern United States. Those are Solanum species in New Mexico and Arizona. And so we hope that this effort continues in 2020 in spite of our pandemic. So most of the presentation has focused on a lot of the service efforts of the NPGS so far, but a lot of the gene bank staff and scientists and curators are also involved in research. And so this is a collage of peer-reviewed publication titles showing the activities of many of the gene bank staff involved in peer-reviewed publications and research focusing on native plant genetic resources in recent years. The two book volumes in the middle is something that I wanted to point out, and that's because Stephanie Greene led efforts along with other editors and other NPGS staff to develop these North American crop wild relative book volumes that are seminal work that looks into inventorying and assessing threats to native North American crop wild relatives. So now what I'd like to do is shift gears a little bit and discuss specifically the Seeds of Success collaboration. And this is a collaboration that was established in 2001 and led by the Bureau of Land Management in partnerships with the Royal Botanical Gardens. And since then, it's an ongoing program with many partners that strive to collect, conserve, and develop native plant materials for restoration and importance in agriculture in the United States. So at our site in Pullman, Washington, Dr. R.C. Johnson, who might be on the conference today, it was a retired ARS research agronomist that was key in initiating the partnership between our site, the Plant Germplasm Introduction and Testing Research Unit, back in 2003. So since then, the SOS program and the NPGS have partnered to conserve and distribute key native plant materials. And as I mentioned, this is mostly implemented at two different sites, the Germplasm unit in Pullman, Washington, which generally incorporates the material into the NPGS, as well as the NLGRP site in Fort Collins that generally, again, secures long-term storage backups, among other things. So the Plant Germplasm Introduction and Testing Research Unit is also known as the Westin Regional Plant Introduction Station. And this is, again, based in Pullman, Washington, on Washington State University's main campus. It was established in 1947, and that year, 1905 in parentheses, is the year of the first acquisitions and material conserved in the gene bank. So some of these accessions are hundreds of years old, or more than 100 years old. The gene bank itself in Pullman conserves about 100,000 accessions, one of the larger germplasm collections within the gene bank system. And aside from the curatorial programs, also has research scientists that support plant genetic resources activities. And to give you an idea about the number of orders distributed, we on average distribute about 1,500 orders of about 40,000 items every year. The curatorial programs in Pullman are divided into 5 different areas. The grasses and safflower, or locally known as the agronomy program. The cool season food legumes, which includes crops like peas and chickpeas and lentils. The temperate adapted forage legume program, which is the one that I lead, that involves crops like alfalfa, clover, and trefoil. There's a horticultural crops with many, many different genera and taxa and species. And there's also a common bean program, or Physiologist program. So when we're talking about the Seeds of Success collections, and as these seed inventories come into the NPGS in Pullman, we follow very detailed processes and very detailed standard operating procedures. So the infographic here, or the flow chart on the slide, just outlines some of those processes. And we don't have to follow it in detail right now, as in the next few slides, I'll sort of break down some of these steps. But in general, the seed is received, and after many different steps, it ends up in one of the final resting spaces, places within the NPGS. So that first process is, as the seed are received, is that we check records for accuracy, and then these seed samples are cleaned if it's needed, further cleaned if it's needed. And then we also obtain 100 seed weights and total seed amounts. We collect digital voucher images that are, again, stored in our Green Global database. And we partition these seed lots into 3 different aliquots if we have sufficient seed. And so if we have 10,000 seeds, then we partition into a 5-degree C distribution sample that stays in Pullman, a minus 18-degree sample that also stays in Pullman as a backup, as well as a second backup sample that is shipped to Fort Collins, Colorado. I mentioned several times already how we manage passport information or associate information to these seed inventories and accessions. And so again, here we have an example of Achnathorum hymenoides, or Indian ricegrass. And what we're looking at is a public-facing Greene Global web page showing information highlighted in the passport tab. And in this tab, you would be able to see information on taxonomy up to the top left, but also some information on who the donor is, and maybe even some narrative information. If you were to migrate to the different tabs here, you would see additional information, possibly some characterization data, as well as digital voucher images like the one you see to the bottom right. One of the other major activities that happens in Pullman is once every year or every 2 years, we make SOS materials available to the Npgs Curatorial programs across the nation. That's because a lot of the germplasm that comes to our site is not a priority for us in Pullman, and could be a priority or important to some of the other gene banks across the nation. So we make a list available of the germplasm that has come in through the SOS program, and curatorial programs and curators make decisions on what germplasm they might want to incorporate into their priority program at their site. Once they've done this, they've made a commitment to long-term conservation of that particular germplasm. And so the 4-degree sample in Pullman, if it hasn't been distributed already to the stakeholders, it hasn't been depleted, and the minus 18 backup sample are then transferred, as well as the records. The Greene Global records are then owned by that priority curatorial program at sites outside of Pullman, but as well as in Pullman. Some of the curatorial programs in Pullman also incorporate some of the SOS materials into their active collections. So that one backup sample that has been shipped to Fort Collins early on does not need to be transferred and stays in Fort Collins. As the materials incorporated into the active curatorial programs, as I mentioned earlier, more than likely at some point will need to be regenerated. And so as seed stocks run low because we are distributing them or because they've fallen below a certain threshold, they need to be queued for regeneration. And the image to the left is of our field regeneration cages in Prosser, Washington for the temperate adapted forage legumes. And we use these insect-proof cages to manage pollinators like the alfalfa leafcutter bee in that inset. We use the alfalfa leafcutter bee, we rear them and use them in the cages, and the cages contain them to help pollinating and seed set in the cages. But also the cages help prevent genetic erosion or potential gene flow between accessions of a given species that could hybridize. What we want to do is these materials that have been collected originally, we want to keep that genetic integrity and prevent any kind of genetic erosion. So the picture to the right corresponds to astragalus, native astragalus that have been collected and incorporated into some of the active collections in Pullman. And we recently optimized some germination protocols for these native species for which no protocols existed. And so since we've regenerated a lot of plants or germinated a lot of plants, we decided that we'd establish them in the field and also begin to optimize some of the regeneration protocols. So I'd be remiss if I didn't take a moment to acknowledge some of the historical research that has been conducted in our site in Pullman. And this is research conducted by retired agronomist R.C. Johnson, mostly on developing native or seed zones working with native plant species. And so the list to the right is some of the empirically derived seed zones that he and his colleagues have developed for many of the different species. The reason I kept that font in red towards the bottom was because this is work that he's summarizing and submitting soon for peer-reviewed publication on the sulfur-flowered buckwheat or Eriogonum. And the pictures in the seed zone map is preliminary information that corresponds to that. So I've also left a marker here to remind myself that this is the point in the presentation that I'd like to turn it over to my colleague, Dr. Stephanie Greene, for her to then talk about some of her activities and focus on about 30 different slides. So Stephanie, we practiced this. Let's hope it works well. Are you ready to go? Can you hear me first of all? And are you ready to go? Okay.
I can hear you. Can you hear me?
I can. And so
Okay.
why don't you just go ahead and start sharing your screen and I'll tell you when I see your slide.
Okay, great. And thanks, Brian, for giving such a good overview of the Npgs and the important work that's being done at Pullman. All right. Can you see my slide?
Yes. And
Okay.
I'll mute myself.
Okay, perfect. All right, so we're going to switch gears and I'm going to talk about what the National Laboratory for Genetic Resources Preservation does in terms of managing the SOS material that we get from the BLM program. So NLGRP is located in Fort Collins, Colorado, and the lab has actually been here since the 50s, when it was recognized that Colorado's low relative humidity actually makes a pretty nice situation for storing seed. And so our role for a very long time has been to preserve and back up plant genetic resource collections under both conventional freezer conditions and also cryogenic conditions. This is namely in liquid nitrogen vapor. So as Brian mentioned, we have quite a large collection. It's approaching 1 million samples that are actually just stored at our facility. And that's because we store the backup collection for the National Plant Germplasm System and also store about 50 different other collections from other agencies. We also do a lot of significant work at our lab, research work. We are developing strategies for exploiting genomic data to enhance how we manage our genetic resources. And also we're developing strategies for sampling, preserving, and using crop wild relatives. All right. Hang on a sec, I'm trying to, okay. So I'm gonna talk then about how we process the SOS seed for long-term preservation. We received the seed from Pullman, and first off, I wanna say that this whole activity is managed by Dr. Christina Walters, and we're very grateful that she has now taken over this process. I think it's bringing an entire new capacity to how we handle these seeds and how we the amount of research that we can focus on native plant materials. So we received seed from Pullman, and the seeds are dried at 5 degrees C and 25% relative humidity. While the seeds drying, we'll take a subsample that we'll then use to assess the quality of the seed. Once the seed is dried, it'll be confirmed to be dry by using relative humidity probes. And then it's packaged in moisture-proof foil laminate pouches and then goes into our -18 seed vaults. So when we conduct the seed quality assessments, what we're doing is we will clean the seed if it needs to be cleaned, and we'll quantify the amount of chaff that's in the seed lot. We then verify the species based on seed morphology and take a look at the maturity, seed fill, pest and pathogen, and mechanical damage that might be impacting the quality of the seed lot. We get 100 seed weight and then seed number, and then we'll conduct a viability test on the seed. This is done either using a germination test or in some instances, we'll do a tetrazolium test. So Dr. Walters has been able to develop a number of different research topics focusing on the SOS seeds that we're receiving. One of the topics she's focusing on is determining what the germination protocols are for these native species. What we're finding is actually there are a lot of species where we simply don't even know how to germinate them. This is important information because it directly impacts our users of the seed lots. I mean, if you don't know how to germinate something, it's going to be pretty hard to get it planted and into the ground. And so Dr. Walters and her team have been working on determining what specific protocols are. She's also been very interested in developing alternative assessment methods, basically focusing on high throughput methods, trying to develop protocols that speed this whole process of assessing seeds and getting them into storage as quickly as possible. Also, during this period of time when the seed's being assessed, can we develop the ability to do rapid phenotyping? So basically, documenting the material, particularly the seedlings, in terms of characterizing their phenotype. And again, this information is something that can be put into Greene that is of value. This information is as valuable as the actual seed itself to have an understanding of what this material actually is. And she's also interested in developing non-destructive tests so that we don't have to use up the seed when we do these testings. And then a final topic that she's focusing on is developing alternative assays for seed aging to get a better handle on when a seed begins to decline in storage. One promising methodologies that she's looking at is looking at how RNA degrades, and can this be used to characterize seed aging? Okay, so I'm now going to shift gears, and I'm going to talk about the Npgs Sos Collection. When I talk about the Npgs Sos Collection, what I'm talking about is that material that's actually coming into the NPGS to be managed and distributed. As of April, we have about 19,000 accessions that we're managing. This figure is a little bit dated. The numbers are from 2020, but what it shows is just the enormous amount of taxonomic diversity that we have in the SOS holdings. We have accessions representing 147 families, 1,000 genera, and 4,000 species. The 10 families that have the most number of accessions are shown to the right, and you can see that accessions in Asteraceae and Poaceae make up over half the collection. So where has this collection been, where has this material been collected? So this heat map shows us that most of the material has been collected in the Western United States, and that the states of California, Oregon, Nevada, and Utah have had a lot of samples collected from them. On the right-hand side is a table that shows the NPGS repositories that have SOS holdings. You can see the repositories as stars on the map. Not surprising, the Pullman, Washington repository has the largest number of SOS holdings, about 12,000, almost 13,000 accessions. That's because the Pullman site has curatorial programs that have native materials in them. For example, the grass collection, and there's also the horticulture collection has horticultural curatorial program has a lot of natives that they manage, as well as Brian's temperate forage legumes. However, Pullman also holds a cohort of about 8,000 accessions that are not actively curated. This material is distributed, and of course, we are backing it up at NLGRP. However, at that point in time when the seed declines, either because it's distributed or the viability has declined, the material will not be regenerated simply because it hasn't been incorporated into a curatorial program. And I'll talk a little bit more about this when I talk about the challenges. I also want to point out that our site in Columbus, Ohio, has the second largest cohort of SOS samples. And this is our ornamental horticulture, our ornamental germplasm collection. So these are basically ornamental plants that they're adopting. So I think it's important to take a look at the Npgs Sos collection from potential agricultural use. Now, granted, all of the material that's been collected from the SOS program has a use of restoration. But what I was interested in looking at is, are there additional uses that this material can be used for? So what this slide shows, well, basically what we did was we went through and looked at the taxa and we classified use based on the taxa. And these classifications came out of John Worsima's book on economic plants. So what we found was that that most of the collection most of the species have a use only for restoration. However, there's a significant number of accessions that have additional uses. For example, there's about 4,000 accessions that are crop wild relatives. There's another almost 4,000 that have forage use, also ornamental use. Food use, these are wild utilized species, so they're probably harvested out in the wild and are used for food. Medicinal use, that's another important set of taxa that are being collected by the SOS program, and also some material use. This would be like industrial use, oils, that type of thing. So in terms of distribution, what type of material is actually being requested from the National Plant Germplasm System? Which of the SOS accessions are being requested? So what we did was, this figure here shows the distribution, the number of seed packets, starting from that point in time in 2006 when we started distributing SOS material. And what we did was we classified the taxa again based on potential use. And what we can see is that about 2/3 of our requests are for species that have use as crop wild relatives, are wild utilized species, in addition to being restoration, having a restoration use. About 1/3 of our seed requests are for species that are used only for restoration. So we can even do a deeper dive and take a look at, okay, what exactly are people looking at when they get an SOS seed packet from the National Plant Germplasm System? And what we found was that the seeds are being used for genetic studies, for botanical and taxonomic investigations, as well as for varietal development. And you can see also that there's a host of other uses being made of this material. I find it kind of interesting that the yellow, which is the fourth most requested number of orders, is actually for historic, cultural, and anthropological research. So it's being used for a wide range of activities, but mainly focusing on genetic studies, varietal development, and botanical and taxonomic investigations. What we've observed over time is that, and this is getting back to those 8,000 accessions that are being held at Pullman but aren't in an active curatorial program, what we're finding is that the SOS accessions are more likely to be incorporated into an NPGS curatorial program if it's a taxa that has uses beyond restoration. It just makes the material more valuable, and curators are more likely to take it on because, again, one of our big challenges is that wild material is in a completely different animal than a domesticated species when it comes to managing genetic resources. So this is, I love this slide because it really kind of demonstrates what the difference between domesticated and wild species in terms of, this is just seed size. So you can see the pistachio in the center of the circle is pretty big compared to its wild counterpart. So what we're finding is that wild collected seed has a lot of quality issues simply because it's been grown in an environment that's less optimal for good seed production. We also find that this wild species, a lot of times we simply don't know the production methods. We don't know how to germinate them. We don't know how to produce seed on them. We don't even know how well they store or for how long they'll store. And then even when we do have a little bit of information, we find across the board that a wild species is just much more difficult to work with. The seed testing takes a lot longer. It's much more difficult to increase seed. We have dormancy issues. Frequently, seed harvest is the frequencies are really low, so we have to do a second regeneration. So it takes a lot of time and effort to manage wild species. So the challenges that we're facing now in the NPGS are figuring out the best way to manage these 8,000 accessions that are not actively curated. They are preserved at Fort Collins and in Pullman, and Pullman is also distributing the material. Our concern is what happens when we start running out of seed. We do have resource constraints, especially at the Pullman site in terms of infrastructure and personnel. And this is one reason why we have so many accessions that we're not able to actively curate. And finally, a big challenge is simply being able to conduct enough research so we can manage these materials in an efficient and effective manner. We need research on seed testing, storage, seed production, genetic diversity assessments, evaluation of the material that will make it more valuable to our stakeholders. We do have some solutions, at least, that we can offer, and one is to really build upon effective partnerships and collaborations within the National Seed Strategy. We also would like to have more funding that could be focused on research and for genetic resource management of wild taxa specifically. I think perhaps one of the best solutions would simply be to have a dedicated curatorial program for native genetic resources. What this would allow us to do is to be able to strategically develop the NPGS native plant collections. What we need to do is manage the content and size of the collection, and it needs to be done in such a way that we can balance user and conservation needs against available management resources. And having a curatorial program would allow us to be able to do this. I think the bottom line that both Brian and I would like to make is that the NPGS can contribute and is contributing and will continue to contribute to conserving U.S. native plant germplasm. However, our efforts need to be in line with our mission and our role, which is one of managing genetic resources that have managing and distributing genetic resources that have value to agriculture, which includes restoration, but also includes other types of agriculture. So with that, I just want to acknowledge, make a few acknowledgments. Of course, none of this work is done in a vacuum. Within the Agricultural Research Service, I'd like to recognize Mike Cashman, Lisa Taylor, of course, R.C. Johnson, Barbara Halier, and I'm not sure who L. Martin is. Brian, maybe you can just chime in there. Must be somebody new. We have got such a big turnover. Are you there, Brian?
I am, but it's not important, so you can go ahead and we'll.
Okay, all right. So all the folks at Pullman who are working on this material, all the folks at NLGRP, that's Chris Walters, her staff, Dan Carver, and the data management team. Of course, the national program staff, Peter Bretting, Pete Batis, and then the folks at our Washington office, our Beltsville office, particularly Karen Williams in the Plant Exploration Office. Finally, there's also Washington State University, and of course, the Bureau of Land Management, Peggy Olwell and her colleagues there. And then with that, hopefully we've got some time for questions.
Absolutely. Thank you both so much. That was very interesting. And I really love all of the pictures and images that you included and the graphs, all very interesting, but just so colorful and fun to look at. I'm just going to check real quick if any questions got posted to the chat. If folks want to raise your hand if you have a question, or feel free to post it to the chat. I found one. Okay. Has there been progress in techniques for storing acorns?
I wonder if that was an autocorrect from accessions, but I'm not sure that we collected many tree species. Maybe let's interpret that as accessions, and Stephanie, maybe you could.
Well, I think if it is acorn, I know that Dr. Walters has been working on acorns. If she's still on the line, perhaps she could directly address the question. Or Lisa Hill, I know she was there earlier.
Yeah, hi. So, well, this is Lisa Hill, and there are an awful lot of people working on preservation of porcus species, and cryopreservation seems to be, the way it's going to have to go, as opposed to conventional storage. But they really are difficult, and it's good that there's such a big group that is working on them, and that nut has not really been cracked yet.
Okay, and thank you. And Christina said she's here, but she has no mic. Okay, and that, by the way, that question came from someone from the California Native Plant Society, I assume CNPS, Andrea. This question comes from Julie Crawford, and it is, what kind of funding would be required for a curatorial program with small amounts of money to look at specific native species be helpful?
Stephanie, I know this is a sort of a tricky question, and do you think that we could, I mean, we can try to answer it, but also we might be able to lean on some of our national program leaders to answer that question. I don't want to put anybody on the spot, but if Peter Bretting wanted to come on and suggest, provide an answer to that question, and if not, we'll field it.
Hey, Brian.
Hello.
Generally, when we would start up a new program, such as we've started for hemp or for coffee, $2 million recurrent, that means every year, would be a good starting point.
So that's good that you answered, because my answer was going to be a little shy of that. So great.
Muted myself. Thank you. Okay, the next question looks like sort of a follow-on to this last one, which is in addition to curation, which is very important, do you think ARS work on empirical seed zones could expand given funding? Seems like this is a large gap.
At least from the curatorial management of plant genetic resources, and that's the mission of the Npgs, I don't think that deriving empirical seed zones is very high for research priorities to us. I think we've proposed or shown that some of our priorities should lie in some of the other aspects of plant genetic resources management, and that might be the empirically derived seed zones might be some research dedicated to other ARS research programs and not necessarily sites that are associated with Npgs. But we're not denying the value of them. I'm just not sure that the Npgs should focus in that area. Stephanie?
Yeah, I agree with you. I think what we've found is that our resources need to focus on the maintenance of these collections. There's an awful lot of work that needs to be done to be able to do an effective job. Doing empirical seed zones, that sort of research falls in line sort of with germplasm evaluation. And again, if you can recall from the slide that Brian showed, that's less of a priority at this point in time for the Npgs.
Maybe I can enlist Dr. Pete Vadas to suggest that maybe ARS outside of the Npgs, other ARS sites that are dedicated to restoration ecology or land, I mean, maybe those sites would be involved in developing seed zones.
Yeah, I think so, Brian. I think it sounds like, we have other locations that, more in line with the land use and that sort of topic. Yeah, as Dr. Bredding said, if anything is new, it does require those new resources.
Okay, thank you all for that. Check out the chat for some more information about oaks for the person who asked the oak question. I would like to ask a question of, I think I wrote down one for each of you, but it could be that it's both of you could answer both questions. So for Dr. Irish, I wanted to ask you if you could revisit or just quickly give me a little bit more information about those in situ reserves. And I may have missed it, whether or not the idea was that those would be on existing public, like forest lands or what that would look like. Do you want to start with that question, or should I put them both out there?
Sure. Let me pull up that slide and look at it again. And this is not one of my areas of expertise. And if Karen Williams is on the call, she could also answer. But yeah, they're proposed in situ germplasm genetic resources reserves, I think is the term that the Forest Service uses. And this is not, again, my area of expertise. It's really hers. And it's a collaboration between ARS, University of Wisconsin, and the Forest Service. And yes, they are on public lands, on USDA's Forest Service lands. But Karen, I would rather you come in and say something too.
Can you hear me?
Yes, I can hear you. So they can.
The Forest Service, along with Carol Spurrier's office and the Plant Exchange office, we developed a few years ago a framework on how we can collaborate on complementary conservation of crop wild relatives, we call it, linking in situ conservation on national forests with ex situ conservation of germplasm in the Npgs. And so this is a general framework on all crop wild relatives. Then the Cranberry Project is a model project to test and utilize that framework, and where we studied populations of cranberries on 15 national forests, and now we're considering the Forest Service has their process of how to especially designate these populations, and we're considering which ones should be designated as what we call in situ genetic resource reserves.
Fantastic, thank you. Very interesting. My second question is, I guess, back on target to you guys, and I thought this might be directed toward Dr. Greene, but I'm certainly interested in input from both of you. I was wondering, it's sort of a two-parter. I like to say two-pronger because that seems a little botanical. What have been the efforts, or and maybe this isn't your office, but to understand the genetic coverage you've got in your collections in terms of, I mean, certainly we may not know the full breadth of genetic diversity across an entire species or population, but I'm curious if there are efforts to look at that to see sort of what aspect, what portion of the biodiversity you may have captured in your collections. And then if you could also answer or talk about whether or not there have been any sort of updates or in the protocols or information on thinking on how to conserve genetic diversity in clonal species, especially those that maybe do reproduce primarily clonally.
So to answer the first question, so that would be doing an assessment of what is the representation of our collections of the actual diversity that's out in the landscape. That's one reason why we need a specific curatorial program that focuses on these natives, because that's exactly the sort of questions that the curators conduct research on, and that's what drives their understanding of the collection, their identification of gaps in the collection, and where it is that they need to go out and make new collections. And so it, other than for the work that R.C. Johnson has done in evaluating diverse material to develop these seed zones, you know, he has done that on a number of species, but there just hasn't been the capacity, because we don't have a curatorial program, to conduct that type of research, which is absolutely critical. If we want a good idea of how well are we conserving these species, this type of research needs to be done. And then in terms of the second question, I can take a stab at that, and then Brian can chime in as well. And I was wishing that Gail Volk, who's one of the scientists at NLGRP, was here as well, because she's done a lot of work with clonal material and how best to conserve it. And actually what we're doing is, especially with native populations, what we're looking at is actually going ahead and collecting seed that's out in the wild. So this is in the case of a species that produces seed but might be clonally propagated. That often for native materials, we can do a pretty good job of by getting a really good seed sample from a large population that represents the diversity that's within the population. It's again, if it's a species that just, you know, doesn't produce seeds, maybe it only clonally reproduce, that's a lot harder option, because again, then you're managing a number of individual actual individual entities, and that takes a lot of resources. You basically have things out in the field, individual trees. And that takes a lot of resources to manage. So if we can conserve them as seed propagales, we're ahead of the game a little bit. Brian, do you have any thoughts on that?
Yeah, actually, maybe you could bring up the point about how source of collecting site, geo-referenced materials, that helps us in incorporating germplasm into the collection if we don't have other genetic information, genetic diversity information. You know, I know that for the natives in particular, you know, there isn't molecular markers haven't been developed, or they haven't been collected widely, or large studies outside of the NPGS haven't been conducted in a lot of these native plant materials. But for cultivated crops is a very different story. But, you know, so gaps in genetic diversity coverage, many times curators make decisions on what to incorporate in the collections based on where they're collected. Not necessarily assuming that because they're collected in different geographic locations, they are genetically distinct.
Yeah, and that's, Brian, the work you've been doing with the documentation in Greene Global. What we're trying to do is, for example, we've been able to pull up, we know where the ecoregions where materials being collected. I mean, we have the latitude and longitudes. So what we can do is extract environmental data. For example, you know, the ecoregion classification, and we can take a look. We also know many of these species have been modeled or could be modeled, so we know what their distribution is, their range. And from that, we can take a look, do some simple tallying of, you know, how many ecoregions does a given species have represented. If we know that X number of ecoregions, we anticipate the species can be found. So that kind of work can definitely be done. Brian has been working with his staff to better document the materials so that these kinds of assessments are a lot easier to make.
That's so fascinating. I could listen to you talking about that all day. I would venture to guess that that mating systems somehow come into play, even if we don't fully understand what all the mating systems are, what the population metastructure type of stuff is. Because the species I'm thinking about specifically is native. It's golden seal, Hydrastis canadensis, and it's patchy. It's, you know, it does reproduce sexually, but that seems to be sort of the last ditch effort. And so there's just these huge clonal patches that, you know, some have been quite large, where they find that they're the same individual. And so obviously with that species, what we've seen is that the genetic diversity is across the populations. So, you know, you wouldn't need 70 collections from within a single population or patch. But yeah, so thank you so much for that. Real quick too, I think Julie Crawford has posted another question that I think is informative to your prior an earlier question. Last week during the CPC meeting, that's the Center for Plant Conservation meeting, they were encouraging everyone to think of how plants could benefit from the Biden administration's funding for climate change. Julie's wondering if this need for curation could somehow tap into the Biden climate funds.
I think we should divert that question to one of the national program leaders.
It'd be great if it did, is all I would say.
Yeah.
We've not seen the details of budgetary details yet. That's of the current administration's first budget. It's not been presented yet, so it's really premature to address that at the moment. In terms of budgets, in terms of the overall climate change adaptation and mitigation, where are the genes? The genes for this are going to be in the gene banks, or they'll be in reserves or protected lands. The genes for adaptation and for mitigation. It's not a budgetary reply. It's just more a genetic reply.
Okay, well, thank you so much for that information. And I actually was planning on mentioning that the climate change, well, actually, no, another aspect, which I'll do during the Native Plants Roundtable. We're about 10 minutes over for Q&A, which is fine. I'm glad that you generated a lot of interest and discussion. I want to give folks another chance. If you have any final questions for our speakers, you know, please raise your hand or put a message in the chat. I think some people don't actually have access to the chat either. I've seen sometimes with large groups that sometimes the chat, I lose access to the chat sometimes. Okay, I don't see any more questions, but I want to thank both of you so very much for your presentations today. And as many people may know, we do post the PowerPoint as well as the recording on our website. And we'll let everybody who was on this call today, we'll email you with the summary notes from today, which will include the information to access the presentation. So thank you so much. Appreciate working with you and listening to your fantastic insights and information. And I look forward to continue working with you.
Most welcome. Thanks for the invitation.
Thank you. Yes.
All right, so we'll finish up this meeting with the Native Plants Roundtable, which provides an opportunity for anybody on the call to give updates on meetings, events, information, publications. I'll start the process while everybody's trying to think about what they came with by mentioning what I alluded to earlier, which was the America the Beautiful initiative was released recently. There was a joint press release from Department of Interior, U.S. Department of Agriculture, the Department of Commerce, and the Office of the President outlining the America the Beautiful initiative with an initial report detailing a vision for a 10-year locally led voluntary nationwide effort to restore and conserve America's lands, waters, and wildlife. We have a link to that, which we'll try to provide in the chat, but it'll definitely be in the notes. And the report seems, well, one of the lines of the report that I found interesting was that it says the report and the recommendations that follow are anchored in a simple truth. Nature is essential to the health, well-being, and prosperity of every family and every community in America. So I think my main hope is that plants will be considered part of that solution. The report identified 6 priority areas for the administration to focus on, and 2 of them in particular are definitely have a nexus with the work that we're doing, which is supporting tribally led conservation and restoration priorities and creating jobs by investing in restoration and resilience projects and initiatives, including the Civilian Climate Corps. So I think that's really great. And I think, you know, the whole message about native plants, you know, being an answer for so many things, including livelihoods, you know, maintaining ecosystems is really, there's a lot of interest around it. I would just want to make sure that everyone's aware of it and that we're making sure that our actual plants are being thought about as part of this. And I think that I will let it go with that. Does anybody else have an announcement or an update to discuss? I don't know if Gary's still on the line.
Yeah, I just raised my hand.
Oh, sorry, I can't see that. I'm sorry.
It's all right,
Hello.
no worries. I just want to announce again, I've shared this on the PCA listserv, but tomorrow and Friday is the 18th annual Smithsonian Botanical Symposium. It's from 1 p.m. till 3 p.m. Eastern Time, both Thursday and Friday of this week, and it's not too late to register. The focus of the Botanical Symposium is on plant symbioses, looking at how plants rely on insects, vertebrates, bacteria, fungi to survive and reproduce. So there will be 3 speakers each day, and you could register at naturalhistory.si.edu/research/botany, and I'll add that to the chat section. Thank you.
Great, thank you. We had a question posed to the chat, and I'm wondering if this person who I'm about to call on with her hand raised may be able to help with that as well. Carrie, I'm not sure what you were planning to say, so I'll let you take it away and then I'll throw a question out there if you don't hit it.
Hey, how's my sound?
Can hear you now. Thanks.
Okay, great. I just wanted to pop in and say hi and thanks for these great meetings, and also just give an update on the Southeastern Plant Conservation Alliance quickly, if that's all right.
Yeah, that's fantastic.
My lighting's pretty bad here, but let me turn around. Okay, I did give an update a few months ago, but we now have an active website and we're working on additional outreach materials. So I wanted to just remind everyone that our alliance was formalized last year, of course, building upon years of strategizing and networking and partners who are already active in states and across state lines across the region, but that we are dedicated to supporting and bridging gaps between local and national efforts such as this to restore and to prevent and restore the loss of plant diversity in the Southeast. We seek to do this by building capacity, facilitating novel partnerships, and leveraging shared resources to stimulate collective success for our region. So I wanted to let everyone know, and I'll add this in the chat, that our website, we have a new external website now dedicated just to the regional PCA. It's se-pca.org. And we have also developed a really nice outreach flyer. We included that in some correspondence that went to DC to the transition team for the new administration earlier this year, both directly from us as well as through the national PCA and some other key groups. We got to present an update and overview on our strategic planning and development on the CPC meeting last week. And I just wanted to let everyone know that we are here to support efforts from the national down to the local, and just to participate as much as we can, especially with these advocacy components, whether that's reaching out to the general public or to people in the agencies and administrations and things like that. So I'll post a couple links and I'll actually go to the chat now and see which question you were talking about. Oh, highlights from the CPC meeting. It was a really good meeting. Advocacy was a very hot topic, as we all know that it is lately, right? And as some of our speakers from CPC and then Native Plant Conservation Campaign on our Southeast PCA back in November said, we're really in the golden age of plant conservation. We're concerned with the right things. We're here at the right time. And as long as we keep paying attention, I think we'll be able to be part of a really great movement that's continuing to push forward the dial for plant conservation across the nation. But they are going to make those recordings available. So if you were registered for the CPC conference, I think you'll be able to still access the recordings from the conference, but the CPC Rare Plant Academy online training center is available. Even if you're not a CPC member institution, you can actually register for an account on there and you can access recordings from past conferences as well as additional training materials. They have even made available the Southeastern Partners and Plant Conservation Alliance conference recordings for 2016 and 2020 are on there as well. So it's a great resource that I encourage everyone to check out. And I'm sure the 2021 conference recordings will be up there within the next month if they're not already. Thank you.
Great. Did anybody else attend the CPC meeting and wanted to just add to that at all? Or any other questions? Okay, I do have a chat here that I'll read because I'm not sure if it's a question or a statement. Federal, state, county, local, and private partners associated with the Nature Reserve of Orange County, California recently kicked off a native seed cooperative to develop an enterprise system to provide genetically appropriate native seed for restoration and enhancement on over 37,000 acres. Wow, that's fantastic. That's great to hear. I wanted to mention as well, I think we might have glossed over it when we were talking with the PCA updates, but the link to the cooperators page was included in the chat, I believe, and I just wanted to make you aware that, yeah, we do not do advocacy on this in this venue, we have a lot of federal employees who are here, and we're not allowed to be involved in that. But the non-federal committee does have their own listserv that is separate from anything, from the public PCA listservs and everything. So when you become a cooperator with the Plant Conservation Alliance, you are added to that listserv. And I'm not sure how active the list is, but I would assume that's where information like you've heard today might be made available. All right, do we have any other announcements or updates? Okay, well, I would like to just make you all aware of our the July 14th Plant Conservation Alliance meeting, which if you haven't figured out the system yet, it's the second Wednesday every other month. Not a hard one. Please just put that reservation on your calendar. Love to have this diversity of people here. Our next speaker will be Dr. Ellen Damshin, who's a professor at the University of Wisconsin-Madison, and she's going to talk about the role of habitat corridors on plant diversity. She's actually done some really great groundbreaking work, 20-year studies, looking at some of these issues, and that's something we don't find very often. There's not often enough interest or money to allow someone to do research over that time frame, but that's such an important time frame in the plant world. So the easiest way for you to stay informed about our next meetings are going to be if you were to join the PCA listservs. We post our meeting messages over all of the lists. We have about 4 lists that focus on different aspects of plant conservation, and we generally do not put the same messages across every list. So if you're interested in medicinal plants, you should join that list as well, because we will put medicinal plant issues or information over that list versus the general Native Plant Conservation Alliance list. With that, I'd like to just once again thank our speakers, Dr. Irish and Dr. Greene, for presenting to us today. Thanks to each of you for joining us, and I look forward to seeing you at our next meeting in July. Take care.